स्टार्ट किया स्टार्ट है चेक कीजिए हाँ चालू हो गया हाँ स्टार्ट हो गया हेलो हेलो ये बार बस ऑडियो की टेस्टिंग कर लेते हैं हेलो 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 स्टार्ट हो गया हेलो ठीक है सर स्टार्ट कीजिए सर Good morning, everyone. Today, the topic for JC presentation is Neuraminidase-resistance in influenza. Presenter is Rajnarayan, preceptor Dr. Saki. The title of the article is Characterization of Neuraminidase inhibitor-resistant influenza virus isolate from immunocompromised patient in Republic of Korea. This paper is published in the year January 2020. This is from the Institution of Korea Center for Disease Control and Prevention, Cheongju, South Korea. The abstract reads like the background of the study is there is a surveillance system called as Korea influenza and the respiratory virus surveillance system that monitors epidemics of influenza and severe acute respiratory infection, and they identify the mutated influenza virus resulting the drug resistance, pathogenesis, and transmission. The method they followed were they collected the oropharyngeal swab, and these specimens were it's confirmed by real-time RT-PCR nucleated on MDCK cell lines, and the HA and NA sequences, Hemagglutinin and Neuraminidase sequences of influenza virus were analyzed, and the mutations were seen. Then Neuraminidase inhibition and Hemagglutinin inhibition assays were utilized to characterize these isolates. The results are there are two isolates, H1N1 epidemic 0-9 isolate harboring H275Y substitution in the neuraminidase sequence were detected in patients with acute hematologic cancer. These patients, and they have shown prolonged respiratory symptoms, and they were not described the treatment of oseltamivir and pyramidine treatment. Though the neuraminidase inhibition assay, both were found to be resistant to oseltamivir and pyramidine, but not to xanamivir. Hemagglutinin and neuraminidase phylogenetic assay suggested that the two H1N1 PDM01 isolates were not identical, and their antigenicity was similar to that of 2018-19 influenza virus vaccine. And they concluded that the utility of monitoring these influenza-infected immunocompromised patients in general hospitals for early detection of emerging neuraminidase inhibitor-resistant virus and maintaining the continuous surveillance of the patient with this influenza-like illness, and also to keep a check on the spread of these new variants. They characterize virus can inform the risk for the risk assessment and future epidemic and pandemic prevention. They can use this the study. Coming to the background and basics of influenza virus. Influenza virus belong to the family Orthomyxoviridae. The virus is spherical, pleomorphic, helical, nucleocapsid. The genome has single-stranded RNA, negative sense they are segmented. These segments are 18 number in case of influenza A and B and 7 in number in case of influenza C. These segments, then the envelope contains HA, hemagglutinin and neuraminidase. These are the spike-like things. These are called polymerase that project out of this capsid. So there are three types, influenza A, B and C. This division is based upon the antigenic characteristics of nucleoprotein and M protein. I will explain the structure in detail. Influenza A subtype is based upon the hemagglutinin and neuraminidase, and they are about H1 to H18 in number and neuraminidase N1 to N11. Seeing the structure of influenza coming from inside to outside, the core is made up of this nucleoprotein, where you can see the RNA, the segmented RNA. That is very clear in the next picture. I will explain. Then that is followed by the protein, then followed by the matrix layer and the capsid. Then that is followed by a small lipid layer, then lipid envelope. Then it is followed by the peplomers, the spike proteins called hemagglutinin and neuraminidase. If we talk about the core, we have segments. Here there are 8 segments, and the gene products of each segment have their significance. Here the structure here is a bit more clear, with the nucleoprotein followed by the lipid envelope. This is called as then we have, there are two M1 and M2. We have beneath the lipid layer we have M1 layer, and M2 is the ion channel here. Then that is followed by the hemagglutinin and neuraminidase. Gluten is hemagglutinin and this orange is neuraminidase. So the gene products of each segment, they are the segments PB1, PB2, PB8. These segments, they are main components of viral polymerase required for replication. Then this segment hemagglutinin, HA, this is mainly used for viral surface attachment and fusion glycoprotein, and this is a major antigenic component. Then major nucleocapsid structural component, it is over here in P, and it is followed by neuraminidase. This neuraminidase has viron surface glycoprotein with sialic acid cleavage activity. I will further explain about this in detail in the next upcoming slides. Then M1 and M2. As I said, M1 is a matrix protein that is below this lipid envelope. Then M2 is non-glycosylated membrane. This is ion channel. This green thing is M2 channel, that is ion channel. Then NS1 and NS2 are the non-structural proteins. So influenza types. As I said, there are 3 types, A, B and C. A is most significant in causing pandemic and seasonal epidemics. These are antigenically highly variable. Influenza B majorly causes seasonal epidemics. Influenza C just causes mild respiratory. Influenza A virus infects multiple species. Here they include human, avians, mammals, pigs. Pig is the reservoir. Here the avian birds are the ones who have a diverse combination of these segmental genome that have the propensity to transmit to every other species, including the sea and the mammals. Coming to the hemagglutinin, this is one of the spike protein on the surface of the influenza virus. Basically, it's a trimeric molecule with a long filamentous stem and has a globular head. This globular head has a receptor site. It also has a short hydrophilic tail for anchorage. The main function of hemagglutinin is for binding and fusion of virus. This facilitates the virus to enter into the host cell via its attachment to the sialic acid receptor. The receptor for these to bind is sialic acid. The significance of hemagglutinin is they are the major determinant recognized by humoral immune system. That is, they are the main antigen against which neutralizing antibodies are directed. And the application of which is these are crucial components for current influenza vaccines. The vaccines are developed based upon the antigenic nature of hemagglutinin. Hemagglutinin is cleaved into two parts, HA1 and HA2. This cleavage is mediated by serine proteases. The cleavage of hemagglutinin is very necessary for the particle to become infectious. Only after the cleavage, the particle is infectious to humans. So, why this cleavage? This influenza virus normally remains confined to this respiratory tract. This is the significance. Since this cleavage, the serine proteases, happens only in the respiratory tract, this influenza virus normally remains confined to respiratory tract because these proteins, enzymes, the cleavage are exposed only at those sites. Coming to the other papilloma, neuraminidase. This is the second major antigenic determinant. This is a tetramer with a spike, with a star, and it also has 4 active sites. The main activity of the neuraminidase is catalyzes cleavage of glycosidic linkages to sialic acid. This the activity of this neuraminidase is this assists in release of viral progeny from infected cell during birth. So after infecting a cell, the final process is budding out of the infected cell and infect the neighboring cells. This main part of budding out and is mediated by this neuraminidase. Neuraminidase prevents self-aggregation, and this is an important target for antiviral agents. These are the neuraminidase inhibitors. Coming to viral diversity, the viral diversity occurs by two ways: antigenic shift and drift. First, antigenic drift is because of point mutations. These point mutations result in formation of new strain variants. If you see this example, here this is the receptor neutralizing antibodies that if they are already developed in the body with previous infection, these neutralizing antibodies, they block hemagglutinin, and this will hinder binding of hemagglutinin to its respective receptors. But because of this point mutation, antigenic drift, what happens is there is expression of this point mutation over here where the receptor binding site is mutated. These already pre-existing antibodies, now they can't bind at this location. That makes it free for hemagglutinin epitopes to directly bind to these receptor sites. So mutation will alter hemagglutinin epitopes so that neutralizing antibodies can no longer bind. The effect of which causes seasonal epidemics. This one variant predominates for 1 to 3 years. Then after that, again antigenic drift occurs. As a viruses evolve, people immune to the older viruses become more susceptible to neurovirus. Definitely, after 3 years, if there is any point mutation and antigenic drift, the people who are already infected and have antibodies, they can no longer get protection, and they are susceptible to new infection. So this necessitates annual updating of vaccine strain. Coming to antigenic shift. This shift is caused by genetic reassortment. Reassortment is a different thing from recombination. This reassortment is possible because of segmental genome nature of influenza virus. One strain has, like if we think these color coding. Red color has a one strain, this blue color one strain. When these two animals or these two or beings, when they come in contact, when the influenza virus, they may recombine, like reassort their segmental genomes with the other, forming a new strain. This will form a novel subtype, and these are mainly important for pandemics. Is a potential pandemic. If person to person transmission if occurs, then it results in pandemic. So this is relatively infrequent and it occurs in 1 to 10 years. If you can see here this is one strain, the other strain. Okay, then genetic reassortment is happening over here, exchanging of viral strains in secondary host. Then after which you can see a completely new strain develops from these, and there is no cross-protective immunity to virus expressing this is the novel hemagglutinin that is originated. The implications of antigenic shift, that is little or no pre-existing immunity. Potentially it is more infectious, more serious illness among those infected, and it is longer and it occurs in atypical seasons. Coming to laboratory diagnosis of influenza, the application of which is there in the current article. The main samples for influenza are throat swab, nasal swab, both. They are collected and put in a viral transport media and they are transported in ice 4 degrees Celsius. Virus isolation, we can see in MDCK cell line, that is Madden, Darby, Kiran kidney cells. They can also be isolated in embryonated hemp egg, mainly in amniotic cavity. Then amniotic cavity, because it mainly the origin can happen in the first amniotic cavity, then the multiplication and mass production can be done in allantoic cavity. So first in amniotic cavity. Then identification and characterization is done by HA and HI, hemagglutinin assay and hemagglutinations. Then next category are molecular detection by multiplex real-time PCR. The other modes of diagnosis are antigen detection, can be from immunofluorescence assay, immunochromatography assay, ELISA, etc. This is the example of MDCK cell line, that is cytopathic effect CPV influenza viruses in MDCK cell line. This is the unaffected MDCK cells. After infection with influenza, this cell line will show lacing pattern. That is characteristic of influenza. Then the other tests that are used in current paper are hemagglutination assay, HA, and HAI. Basically, hemagglutination assay can be described with like certain virus possesses glycoproteins called hemagglutinin that hemagglutinate with RBCs. Here in the reaction, you can see the reaction of viral hemagglutinins with RBCs result in lattice of agglutinated cells that settle irregularly in the tube, microtaper of in a tube or in a microtaper. The hemagglutinins of a virus will react with RBCs and form a lattice of agglutinated cells, and they get settled in the microcatheter, or if it is done in a tube, it will settle at the bottom of the tube. Unagglutinated cells settle in compact bottom. This process is known as hemagglutination. Hemagglutination occurs when influenza virus and RBCs are mixed. Then coming to the titer of hemagglutination is the reciprocal of the dilution of virus in the last well with complete hemagglutination. Here the dilution is made, and the last well with the highest dilution of this virus, that showing the hemagglutination is the HA titer. So the RBCs that are used in this procedure are for season influenza, human blood group O RBCs are used, and guinea pig RBCs are used. The percentage of RBC is 0.75%. For avian influenza, we can use chicken or turkey RBCs. The concentration is 0.5. Then what is hemagglutination inhibition assay? Here, if the serum of a person infected is mixed with RBCs and the test virus, there won't be any agglutination of RBCs. The meaning, if a person is infected, he has developed antibodies against, for example, influenza. He has influenza antibodies in him. Then a mixture of RBCs and test virus. RBCs and test virus, if it is mixed with the antibodies present in the serum of infected person, these antibodies will react with this test virus and they get neutralized, and remaining are the free RBCs. This the explanation of is written here. This arises because antibodies present in the serum of infected person reacts with the test virus and neutralizes them. This is positive. In case if the patient's serum doesn't contain antibodies against the surface proteins of test virus, then there will be presence of hemagglutination, as there are no antibodies. So these viral hemagglutinins will react with RBCs and hemagglutinate the RBCs. So finally, in hemagglutination inhibition, the antibodies present in the serum of the infected person, if it is mixed with RBCs and the test virus, these antibodies will react with the test virus and neutralize, and only RBCs are left. That shows that they are free RBCs are left. So here HAI is performed by determining the highest serum dilution that inhibits RBC hemagglutination caused by viral hemagglutination. Here, this is the highest dilution. If the highest dilution shows that inhibit the hemagglutination, that titer is taken as HAI. Coming to management of influenza, mainly supportive care given by acetaminophen, that is PCM, and about aspirin for children. The antivirus used for influenza are neuraminidase inhibitors like oseltamivir, Tamiflu, zanamivir, peramivir, and laminavir. The other categories are inhibitors of endonuclease activity of viral polymerase. These are CAPS. The examples of which are paloxavir, marboxazine. The other category are inhibitors of M2 channels, that is adamantines, that is amantadine, rimantadine. The article proper will be discussed by Rajnarayan.
Thank you, sir. Good morning, everyone. Today the GC topic is Neuraminidase-resistance influenza virus. This research article is characterization of Neuraminidase inhibitor-resistant influenza virus isolated from immunocompromised patients in the Republic of Congo. So background. Neuraminidase inhibitors are globally utilized for the treatment and prevention of influenza type A and B infection. Neuraminidase inhibitor suppress the action of neuraminidase on the surface of the virus to prevent the spread of poisoning virus from infected cells. Oseltamivir, peramivir, the most commonly used drugs for patients with influenza-like illness, helps to relieve clinical symptoms within 2 days of symptoms manifestation, shorten the virus release period in the respiratory tract. However, since most recently detected type A influenza viruses harbor the resistance variation S31 in the matrix gene, and M2 proton channel blocker amantadine and amantadine are no longer clinically applied. The Korea Center for Disease Control and Prevention (ECDC) operates the Korean Influenza respiratory viruses relay system to monitor epidemic features of influenza viruses and analyze virus characteristics, including drug resistance. If a drug resistance mutation is found in the antigen, this KINRESS attempts to isolate the virus and perform phenotypic analysis. That is an inhibition assay. Neuraminidase inhibition assay. Neuraminidase inhibitors resistant viruses were identified in cases in which influenza virus was continuously detected following neuraminidase inhibitors treatment of hospitalized immunosuppressed patients rather than in clinical outpatients. Here the drug resistance is H1N1 pandemic. This is 2009. Viruses were detected via the KINRESS in patients with acute hematologic cancer not exhibiting recovery despite Tamivir and Tenamivir administration. These were characterized genetically and antigenically following isolation. Some the methods are like clinical specimen collections. So how we do collect this specimen? So oropharyngeal swab samples were collected from persons with influenza-like illness. So the KINRESS and processed in the national standard laboratory. Swabs were stored in viral transport media until further analysis. This from this sample we extract RNA. This viral RNA was extracted from 140 microliter of sample, medium using the 2IM viral RNA kit, and according to this kit is according to the manufacturer's instruction. This one-step multiplex real-time PCR in real time from transcription polymerase chain RT-PCR was carried out using the GPAC ID one-step RT-PCR system, and table 1 listed multiplex this timer and tap probe sets. 20 microliter reaction mixture contained 5 microliter of RNA template, 590 nanometer timer, and 140 nanometer probe trail microliter of two times reaction buffer point, 8 microliter of enzyme mixture and RNA free water. This RT multiplies 002 PCR performed on UBI is 7500 fast instrument. This was applied biosystem with thermocycler condition for the transcription, reverse transcription 50 centigrade for 30 minutes. Hot start DNA tag polymerase activation at 95 centigrade for 10 minutes, followed by 40 cycles of denaturation. This denaturation 95 centigrade for 15 seconds, and annealing or extension. This annealing at temperature 55 centigrade for 20 seconds. Then after this data acquisition and analysis of this real-time PCR, assay performed using STS software version 1.4, which is applied by systems. And this table 1 is given primer and probe sequence information to detect how influenza virus type A and B are subtypes, that is H1 and H3. This here type A, B, H1, H3 and C. This here sequence is 5 direction is 5 to 3 direction, and this here target in base pairs. This is all are heterogeneous bases here, and this is in forward in forward sequence and this reverse sequence. As far as isolation in MDCK cells lines, this MDCK cells line cells mostly these cells were seeded at 105 per ml in T25 flask with this flask with Dalbecos modified eagle medium. This medium is high clone 1 BT USA containing mostly 0% fetal bovine serum, high clone, and 1% penicillin streptomycin in a final volume of 5 ml and incubated at 37 degrees centigrade in a humidified atmosphere with 5% CO2 for 48 hours. When these cells reach 90% influence culture medium, discarded and cells were washed 3 times with non-phosphate and multiple of 2 buffered saline. Then 0.2 ml of clinical specimen added into a flask to allow inoculum to absorb 30 degrees centigrade for 16 minutes. And medium, this is 5 ml containing 2 microliter per ml TBCK trypsin added to the flask. And the culture observed daily for cytopathy and morphological changes using an inverted light microscope. This culture supernatant harvested when 75% of infected cells showed cytopathic effect and stored at -70 degrees centigrade. Finally, E. coli, this S002 2019 and 003 2019, AH1N1 pandemic in 2009, viruses isolated. Then Hemagglutinin and Neuraminidase sequencing, how this sequencing can do. And viral RNA was extracted from oropharyngeal swab sample and their isolates using the YAM viral RNA kit. This was poison according to the manufacturer instructions. Reverse transcription performed to obtain copy DNA using the U12 primer. Then Hemagglutinin neuraminidase genes were amplified with the specific primers. This PCR product purified using the KVP, this PCR purification kit according to the manufacturer's instruction and subjected to direct sequencing using peptide terminator. CV for 3.0 cycle sequencing ready reaction kit. This is applied biosystem together with primer for each PCR fragment. And these are how we can do this sequencing or this process is given at level 2 on the API 31 Excel genetic analyzer automated sequence. Nucleotide sequences edited, assembled and aligned using Mega this software 10.1 software and complete hemoglobin conducted using Kushwaha and phylogenesis were constructed using the Mega joining method and bootstrap 1000 times with this Mega 10.1 the software using HA multiple of 0.2 times neuraminidase line DNA sequences of this current influenza AH1N1 pandemic 2009 reference vaccine and other isolates from the Republic of Korea. This table is given how the sequencing primer for Hemagglutinin and neuraminidase of this strain H1N1 pandemic 2009 viruses. So neuraminidase inhibition assay is defined as fluorescence based neuraminidase inhibition assay conducted using the neuraminidase from influenza neuraminidase circuit. This is made by Applying susceptibility of influenza viruses to neuraminidase inhibitor characterized using acetylcholine, zanimivir, and peramivir at concentrations that limited neuraminidase activity by 50%. As defined by WHO, influenza A viruses with normal reduced and highly reduced neuraminidase activity inhibition exhibited less than 10 folds, maybe 10 to 100 folds, and maybe more than 100 folds increase in IC50 respectively. Last considered is clinically resistance. Hemagglutinin inhibition assay. Assay performed according to a standard method. 4 hemagglutinin units per 25 microliter of strain A H1N1 pandemic 2009 virus tested using 0.5% suspensions of the red blood cells. HI titer, reciprocals of highest dilation of sera that limited hemagglutination. Post-infection, net anti-sera against 18-19 and 19-20 vaccine viruses treated with receptor destroying enzymes. After this result, this strain H1N1 pandemic 2009 detection and isolation in immunocompromised patients. Influenza A virus detected in only fever swab samples collected from patients with lymphoblastic leukemia. Patient A is a female 37 years old and relapsed lymphoma. Patient B, female 38 years old with real-time PCR and a CT value of 28 and 29 respectively. Strain H1N1 pandemic 2009 viruses identified by additional H1 subtype detection respective CT value of 28 and 29. They are given table 3 and next were observed within 48 hours following a specimen inoculation in MDCK cells with harvested supernatant exhibiting an hemagglutinin titer of 256. The isolates were named this A Korea S002 2019 and S Korea A Korea S003 2019. This table 3 given the strain A H1N1 pandemic 2009 virus section in immunocompromised patient by real-time PCR. Where patient is patient A and B. Genetic detection using real-time PCR. Then genetic characterization is neuraminidase in genetic analysis for screening drug resistance, phylogeny, and variations. So here given showing that neuraminidase genetic mutations compared with this different 2018 N1 and where viruses amino acid identified with this strain here given. And in Korea, the two strains 003 and 002 strains, and how this 90 amino acid identified in 98.3 and 98.5. And here phylogeny is a tree of neuraminidase gene of drug resistance. This pandemic 2019 is here given. Then neuraminidase, this hemagglutininin, phylogeny, and their variations. Like this table showing hemagglutinin genetic variation compared with this two patients here A and two was this strains 003 and 002 2019. And where phylogeny tree is shown here. Vaccine here dark black is vaccine virus. This light characterization reference virus, and this Neuraminidase inhibitor-resistant virus. So this phylogeny we can see here. Last discussions Neuraminidase inhibitor-resistant strain A H1N1 virus having neuraminidase gene H275Y variation first identified in 2007 and frequently identified worldwide in 2008-2010. However, since 2009, this strain H1N1 pandemic 2009 it occurred in less than 1% of H1N1 pandemic 2010, mainly reported in patients with impaired immune system due to this two times suppressive therapies for conditions including cancer and organ transplant. Because this person suffering from cancer and organ transplant, that patients reduce this suppress immunity by more than two times. Notably, such immunopatients suppressed by more than two times, compromised patients when infected with influenza virus could spread virus over a long period like causing increased transmission. Moreover, as they said, the drug resistance virus while maintaining transmissibility. These patients may constitute a source for spreading drug-resistant influenza in the community. So here, neuraminidase inhibition assay for strain influenza A strain H1N1 pandemic 2009 viruses are isolated in immunocompromised patients suffering from cancer, TB, like so here showing. Mutation suspected to represent antigenic drift in immunocompromised patients were more likely to be observed in neuraminidase than hemagglutinin, peak at very low frequency around 1%. However, neuraminidase mutations did not affect antigenic properties besides drug resistance, although hemagglutinin and 158T substitution increases the binding affinity of strain H5N1 avian influenza virus and human receptors alpha 2 6-linked serum sites. Function of this Korea 0AS002 2019 hemagglutinin A158E substitution functionally remained unknown, but it was not involved in antigenicity in this study. Alternatively, neuraminidase and hemagglutinin have different mutation spectra in swine influenza viruses collected from April to June 2019 in the United States. The antigenic drift in immunocompromised patients may allow swine influenza virus insertion to occur without pig-to-human transmission, ultimately reducing the efficacy of influenza vaccine scheme. Anti-sera from food immunized with the influenza vaccine exhibit good inhibition of this Korea S00 2019 and S003 2019 vaccine. Here, hemagglutinin inhibition assay virus hemagglutinin limited titer post-infection fluid sera seen in Brisbane and Kiran. So, last conclusion we is neuraminidase S20 275Y substitution and some neuraminidase subtype multiple of two types situation in drug resistance influenza A strain H1N1 pandemic 2019 viruses isolated from immunocompromised patients influence both antigenicity and neuraminidase inhibitor resistance. Viruses showed highly reduced inhibition with over 100 folds increase in the IC50 for oseltamivir and peramivir, but not zanamivir. Zanamivir might be option for immunocompromised patients when oseltamivir and peramivir are not effective. These findings also indicate necessity of monitoring for Neuraminidase-resistance viruses in outpatient testing clinic and inpatient, particularly for immunocompromised patients with severe acute respiratory syndrome in general hospitals. Additionally, virus characterization can facilitate the risk assessment of neuraminidase inhibitor viruses. Thank you. Thank you very much.